Hello, my name is Beatriz Vercesi, and I'm a master's student at the University of São Paulo, Brazil. My work is about identifying regulatory epigenomic changes that can be associated with isocitrate dehydrogenase 1 and 2 mutations across different cancer types using bioinformatics tools. IDH1 and IDH2 mutations have a frequent occurrence in some cancer types such as gliomas, and the occurrence of these mutations can lead to alterations in the DNA methylation levels in tumor cells. It has also been related to prognosis in some cases, but the regulatory epigenetic mechanisms behind their occurrence still remains an unexplored topic of investigation. Our objective with this work is to identify these epigenetic changes that are associated with IDH1 and IDH2 mutations across different cancer types. Initially, we will identify the probes that are different methylated in tumor samples harboring these mutations compared to IDH wild type and control samples of the same cancer type. For this analysis, I used R language in the RStudio environment. First, it was made the download of DNA methylation data from the CGA database of 9 cancer types. These cancer types are the ones that showed the higher occurrence rates of IDH1 and IDH2 mutations in the CGA database. Next, as shown in the figure number 1 in my poster, I explored the DNA methylation data using principal component analysis to search for possible similarities in the DNA methylation patterns across the different cancer types. Following the exploratory analysis, it was made a different methylation analysis using as method the Wilcoxon test. For this step, it was made a comparison between ID8 mutant tumor samples and ID8 wild-type samples of the same cancer type as shown in figure number 2. For the final step, it was made the identification of specific methylated probes in ID8 mutant tumor samples for each cancer type, following two analyses: ID8 mutant tumor samples versus ID8 wild-type tumor samples, and all tumor samples versus control samples for each cancer type. The cancer types were analyzed individually with the exception of lower-grade glioma and glioblastoma that were analyzed as a group. The results showed that the ID8 mutant tumor samples have a higher number of hypermethylated probes when compared to the ID8 wild-type and control samples. In conclusion, we can propose that IDH1 and IDH2 mutations led to a hypermethylator phenotype in cancer, and that these mutated tumors have a specific pattern of DNA methylation when compared to wild-type tumors. Other analyses are now in progress to evaluate possible altered targets and the clinical relevance of our results.